Let's begin to discuss the muscular system. We've already looked at the muscular system in terms of muscle tissue, and so we'll take a moment here to compare the three types of muscle tissue as an extension of what we discussed during the unit on tissues. So to start us off, we'll look at skeletal muscle tissue. And so we can see that the location of skeletal muscle tissue is that it overlies the skeleton. It attaches to bone, hence the name skeletal muscle. It attaches to bone, and then as it flexes, it allows us to move those bones around joints, causing movement. If we zoom in on the cellular appearance, we can see that skeletal muscle is multinucleate. It has many nuclei per cell, and it is also striated in appearance, meaning that it has the lines that are running opposite to the direction of the cell itself. In addition, the cells are cylindrical in shape. They look like cylinders or long rods. And so one other thing that we should note is that skeletal muscle is voluntary. So I decide to move my skeletal muscles for the most part. Of course, sometimes we have reactions that we don't think much about, but for the most part, my skeletal muscle is voluntary in action. We can then compare this to the second type of muscle tissue, smooth muscle. We can see that smooth muscle is found in organs or viscera. Those are two terms meaning the same thing. For example, the stomach and the intestines shown here. And so these are our internal organs and any muscles that allow them to move. So again, for example, the muscles in our intestines that allow us to move food through our intestine are smooth muscle. Smooth muscle has a spindle shape, meaning, as you can see at the top here, it's got a point at either end and a little bit wider in the middle, and it is uninucleate. There's one nucleus per cell. They are non-striated, and then they are, as mentioned previously, involuntary. The third type of muscle tissue is cardiac muscle tissue, and we find this in only one place. The term cardiac is referring to the heart, where this muscle is located. We can see from the image at the top that cardiac muscle is striated, though not as dark as in skeletal muscle, and it is uninucleate. It has one nuclei per cell. One thing we should note about cardiac muscle as well is that it branches. So it is not spindle-shaped, it's not rod-shaped, though it is somewhat cylindrical. There are branches where the muscle seems to sort of split off into two pieces and then connect to multiple cells. The other thing we should note is that where the cells connect, we have the plasma membrane between the two cells, but within that plasma membrane we have tight junctions, which are also called intercalated discs. These intercalated discs, or tight junctions, have protein holes, protein passages that allow materials to move from one cell directly into the other. And so those materials are actually ions, sodium ions, that allow for the transfer of the contraction signal from one cell directly into the next cell. And ultimately, we can look at that and think about it in terms of how our heart functions relative to our other muscles. Our heart functions via contractions of the heart to pump the blood out, and so we actually have one muscle contracting, or one muscle cell contracting, and then we want that to pass directly into the other cells of the heart, of the muscle tissue of the heart, because we want the signal to quickly spread throughout the heart and pump the blood. Within the skeletal muscle and the smooth muscle, the individual cells are actually being triggered by nervous signals. In this course, at various points, we're going to look at smooth muscle and cardiac muscle. So smooth muscle when we get to things like the digestive system, and cardiac muscle when we get to the heart. But for now, we're going to focus on the anatomy and major functions of skeletal muscle. And so let's take a closer look at a skeletal muscle. So here we have a skeletal muscle similar to the image on the left, where we have the arm with the bicep on the one side, the left side in this case, and then the tricep, which is shown that it is actually cut or dissected here so that we could look at the other end of the tricep. And so we can see that the muscle is attached via tendon to the bone. And then through the cross section of the muscle, we can notice a couple of different layers here. So we can see actually bundles of tissue inside the muscle organ itself, the entire tricep. And so these bundles, as are indicated on the right side here, are fascicles. And so we have many fascicles making up the overall muscle. Around the outside of the complete muscle, we have the epimysium. Remember that prefix epi means above, and then mesium is referring to muscle. And so the epimysium wraps around the complete muscle. As you can see, it has in brackets there the fascia. This fascia, this epimysium, is continuous with the tendon. It actually runs around the outside of the muscle and then forms into the tendon, which attaches the overall muscle to the bone. And so there's this continuous structure so that as I flex, the fascia gets pulled on, the tendon gets pulled on, therefore. Around each of the fascicles, we have the perimysium. So peri, the prefix means perimeter. So in this case, it's the perimeter of the muscular fascicles. And then within the fascicle, we have individual muscle cells. And around each muscle cell, we have the plasma membrane, which we'll come back to in a moment, just like any cell. But around that, we have the endomysium. Remember that prefix endo means inside, and so it's referring to inside the overall muscle, but around each individual muscle cell. And so we have the muscle cell that's been pulled out in the image here, and it's actually a cross-section as well where we're looking inside the muscle cell. So there's a couple of terms here to note. So we have a cell membrane, just like we do in any cell. We learned that earlier in the course. But in muscles, we call it the sarcolemma. The prefix sarco is referring to muscles, or muscle tissue. And so we have this different term, sarcolemma, but that just means the cell membrane of a muscle cell specifically. We also have the overall term for a muscle cell, that is muscle fiber. We call it this just because when you look through a microscope and you're looking at these cells, as we mentioned earlier, they are cylindrical or rod-shaped in appearance, and so they look like fibers running through the entire muscle. And so we use the term muscle fiber as well as muscle cell, and they mean the same thing. Some other terms that we should note are the T-tubules. The T-tubules, the T here stands for transverse. We learned earlier in the course that transverse meant to run across. And so the T-tubules are little tubules that run across the muscle cell. They're actually made up of the plasma membrane, the sarcolemma, that forms into a tube, and then the tubes, the T-tubules, run through the cytoplasm to the other side of the plasma membrane, or the sarcolemma. On the inside of the cell, connected to the T-tubules but separated by a membrane, is the sarcoplasmic reticulum. Sounds like a mouthful, but again, the sarco is referring to muscle cells, and then the plasmic reticulum is referring to the overall tubular structure, just like we looked at the endoplasmic reticulum earlier in the course. In this case, it's a modified endoplasmic reticulum, but found in muscle cells, so it's a sarcoplasmic reticulum. The sarcoplasmic reticulum, as is indicated in the image, is a place where calcium is stored within the muscle cell. We'll see in a moment that the calcium plays a role in triggering the muscle contraction. Throughout the cell, then, we also have myofibrils. We actually have one myofibril pulled out of the cell here, but you can see that the cell is just packed with different myofibrils running all up and down the muscle cell, or the muscle fiber. Earlier we spoke about the striation of skeletal muscle, this repeating pattern of lines in the skeletal muscle when you look at it under a microscope. The reason for this is because of all the fibers that are running along and making up the myofibrils. And so if we look at one region of the myofibril here that is of importance in understanding contraction, we look at the sarcomere. And so we can see that the sarcomere extends from the one line encircling the myofibril to the other line. But the other thing we should note is that there's a repeating sequence of sarcomeres. All up and down the myofibrils making up this muscle cell are sarcomeres repeating over and over again. And we can see at the bottom here the structure of the sarcomere. For now, we won't worry about the relaxed versus contracted, but I will say that ultimately the sarcomere is the contractile unit of the muscle. And so, to look at the relaxed sarcomere, we can see that there's a Z line on either end of the sarcomere. This is made up of proteins, which names we will not worry about. But then we have two fibers that we do need to think about. And so we have a thin myofilament, which is actin, and we have a thick myofilament, which is myosin. You can see that at either end of the thick filament, the myosin filament, there are myosin heads that stick out off the main portion of the myosin protein. And so what ends up happening is, in order to contract the sarcomere and the overall myofibril and the overall muscle, is that the myosin heads will attach to the thin filament, to the actin, and then they will shift the actin towards the center of the sarcomere. And so if we look on the right side, we can see the contracted sarcomere because the myosin heads have attached to the actin and have actually shifted the actin towards the center of the sarcomere. The Z lines have actually gotten closer to each other. The sarcomere has contracted. Hence, as I said earlier, the sarcomere is the contractile unit of the muscle. Now we've had an opportunity to describe the anatomy and specifically the microscopic anatomy of a muscle. And we've talked about the major functions of contraction within a muscle and within the sarcomere. But now we want to take a step back and look a little more at the gross anatomy, the large anatomy, and describe the tendons and use the terms origin and insertion to talk about specific tendons that are connecting ultimately the muscle to bone. So if we were to zoom in here, we could take a closer look at the muscles of the upper arm, and we can see the bones that are involved, the muscles that are involved, and then that there are origin tendons and insertion tendons. Let's take a closer look at the bicep. So we can see that the bicep actually has two origins. The definition of an origin are that these are the tendons that are attached to a bone that does not move when our muscle makes an action. The definition of insertion is the tendon that is attached to the bone that will move when we undergo a motion. And so we can think about flexing of our arm. As you flex your arm, the bones of your forearm, in this case the radius is where the insertion tendon is attached to, will actually move towards your humerus. In this case, the origin, which attaches up at your shoulder girdle, is actually not moving at all as you flex. And so because those bones of your shoulder girdle do not move, we call the tendons that attach to them the origins, versus the insertion, that is the point of attachment on the bone that is moving in a motion. One thing to note is that the bicep brachii actually has two origins. And so in this course, you're taking a look at specific muscles that you will need to remember for tests and quizzes and so on. But a little trick in terms of strategies for remembering names is that these names were created by biologists in order to help describe certain aspects of them. And so in this case, the biceps brachii, the bi in biceps is referring to two. It has two origins. The brachii is from the Latin term that means arm. So now let's take a look at the tricep brachii. And let's discuss where the origin would be located and where the insertion would be located. Before we do, we can talk about the term tricep brachii. Again, this is referring to brachii means arm, and tricep is referring to that there are actually three origins for this muscle. Not all of them are shown in this image here, but there are three origins. Where would they be found? So the origins for the tricep, as I use my tricep to extend my arm, the portion of my arm that is not moving, again, is my shoulder girdle, my scapula, and the places where these origins are attached. And so the origins are at this end of the tricep. When I extend my arm, the portion of my arm that is moving are the bones of my forearm, and so my tricep brachii has an insertion tendon that runs down. It actually runs around my elbow and attaches to my forearm bones. We can use these strategies to think about the origin and insertion for any muscle which we might be discussing, because we can analyze the motion that is being created by the muscle, and then we can analyze which end of the bone is actually moving, and that's where the insertion attaches, and then which portion is stable, and that is where the origin or origins are attached. Taking a look at this same muscle, we want to define the terms prime mover, antagonist, and synergist. And so if we were to think about flexion of the arm, so if you were flexing your arm, we'd actually have that the bicep brachii is the prime mover, which means, just like it sounds, the prime meaning main and mover meaning the muscle that's actually causing the motion. So as I flex my arm, the bicep brachii is the prime mover. Another term for that is agonist. And so it's one that you might not be familiar with, but it helps us to understand the second term here, which is antagonist. As I flex my arm, my bicep is the prime mover or agonist, and the tricep on the other side of my arm is relaxing, not flexing. And so it is the antagonist. The prefix ant or anti is meaning against, and then referring to the agonist, so it is against the motion of the agonist or the prime mover. And so as I flex my arm, my bicep is getting shorter and flexing, but my tricep is being stretched out. It is the antagonist. It will cause the opposite motion. One thing that we should note is that as I extend my arm, these roles switch. So as I extend my arm, it is my tricep that's causing the extension, and therefore it is the prime mover or the agonist. And then the bicep is being stretched out as I extend my arm, and so it is the antagonist muscle. And so don't memorize these muscles in terms of the bicep brachii is always a prime mover, tricep always an antagonist. But again, we have to think about the motion that's being caused, and then we have to analyze which muscle is causing that motion, that is the prime mover, agonist, and which muscle is being stretched or is against that motion, and that will be the antagonist. Moving on to the term synergist, there's actually not a synergist found in this image here. But the prefix syn kind of means together. A synergist is a muscle that is a helper muscle that helps the same motion. And so we find these throughout our body, but they tend to be very small. For example, at the back of your knee, you have a muscle, the popliteal muscle, which acts as a synergist to help in the flexion of your lower leg. However, the main muscle, the agonist that causes flexion, is the hamstring muscles. And so the synergist is just a helper muscle that causes the same motion, but if it was the only muscle causing that function, our joint would not contract as it normally does.